So good afternoon, everybody. My name is Gauthier Schang, and I'm a PhD student working in Dr. Daniel Bernard's lab at McGill University in the Department of Pharmacology and Therapeutics. And I will be attempting to do an overview of one of my research projects, which involves understanding how follicle-stimulating hormone is produced. Now, what is this hormone and why do we care? So fertility in general is under the control of two major hormones, luteinizing hormone and follicle-stimulating hormone, both of which are produced by the pituitary gland at the base of the brain. Now, both of these hormones have essential functions in the testes and the ovaries, and this is what allows fertility and reproductive potential. Now, both of these hormones have a common alpha subunit. However, their biological activity and their receptor selectivity is determined by the specific beta subunits, LH beta and FSH beta. And so we're trying to understand exactly why gonadotrope cells, which are the cells in the pituitary gland secreting these hormones, are the only cells able to actually produce them. So to try and understand and answer this question, if we first look at the LH beta subunit, it's quite well understood how this is produced by gonadotrope cells. So here I'm showing you a schematic of the LH beta promoter, and we know that three different transcription factors that are listed here are absolutely essential for transcription of this gene. We know that all of these three transcription factors must bind the promoter, and this is what allows transcription of the gene. However, what about FSH beta? Do we have as clear of a picture for this gene? However, we do not have such a clear picture. We do know that ligands belonging to the TGF-beta superfamily are signaling in these cells, and we know that there are triggering intracellular signaling cascades, and this is our best understanding of what is happening at the level of the nucleus after this signaling occurs. We know that different SMAD transcription factors have to bind the FSH beta promoter, as well as the FOXL2 transcription factors. However, SMADs are ubiquitous proteins, and FOXL2 is expressed in other cells in the pituitary gland, as well as cells in the ovary. So while we know from in vivo and in vitro studies that these transcription factors are essential for FSH beta transcription, they are not enough to explain the selectivity of FSH production by the gonadotrope cells. And so the aim of this project is to use in silico, in vitro, and in vivo approaches to identify novel transcription factors that allow this specific expression in the cell type. And ultimately, this will allow us to identify novel targets for therapeutic approaches for human infertility. These are all the wonderful people working with me in the lab, as well as all our funding agencies. And thank you.